I'm Lara Szewczak, scientific editor at Cell, and I'm here with Bernard de Massy from the Institute of Human Genetics in Montpellier. Hi.
Hi.
How are you?
I'm fine, I'm great, thank you.
Yeah, so your lab focuses on meiosis and the process of recombination, and I'm curious, what got you into the field in the first place?
I started to be, the first place my interest was evolution, and then I got trained into really DNA metabolism, DNA replication, DNA recombination. And then I realized that there was one area where I could really try to develop, to understand molecular mechanism, but with their evolutionary implication. It's the process of recombination during meiosis, because it's a process, it's really a molecular machinery, but which has very long-term consequences on transmission of genetic information and genome diversity. So that was, for me, that was really what made me choose this field.
Okay,
yeah.
and what was the question that you started with when you started working on meiosis?
The first question was to characterize, so that was on the budding yeast, and that was to characterize some specific sites of meiotic recombination in the yeast genome, because they were just being discovered just a few years before that. So, and then I've been really continuing this trajectory from yeast, but now, well, now since already 20 years, I shifted to mouse, and similar question. Then I challenged in mammals, so in mouse meiosis.
And so the talk that you gave earlier,
yes,
you were really focused on how the breaks form. So tell us a little bit about that.
So there's, I was in this last year's project with respect to the question you're asking and this molecular mechanism and evolutionary implication. We discovered 7 years ago a factor, PRDM9, and I think it's almost a dream for me, because it turns out that this molecular, this protein, is at the same time, by understanding it, we characterize the molecular mechanism of breaks that are induced in meiosis, and very unexpectedly, actually, it turns out to have enormous evolutionary implication. So what I presented just 2 days ago was to identify how this protein works, and there are other aspects which I didn't have time also to present, which is the evolutionary implication of the way this protein works. So this protein works by specifying the sites in the genome where recombination takes place, and it's amazing, because it really breaks some established concept about the genetic maps, which were genetic linkage, which were thought to be, fixed and following some rules according to, I don't know, gene function, for instance. And this gene, the way it works in mammals, shows that the genetic map is incredibly plastic.
So let's talk about first, how does it work?
So the protein, it's a protein, this protein in particular, so that's one of our projects. It binds to DNA. Then it modifies histones, the histones that are on the nucleosomes adjacent to its binding site. So it has a specific DNA binding recognition. But we evaluate it binds to, maybe it has 10,000 potential sites in the genome, 15,000 potential sites in the genome. So it modifies the nucleosome, the histones, and then we don't know very well actually.
Oh,
so then
okay.
we're working on we assume that it interacts with other proteins, and I presented yesterday a protein with which we think it interacts to, and then again there are some postulated interaction we are currently trying to validate to attract the protein that catalyze the formation of double-strand breaks, and the catalytic activity. We know which protein encodes for the catalytic activity. It's SPO11, but we don't know how these proteins get together. So that's this part we don't know, or we have some prediction, but we don't know how it works.
So one thing I found surprising about your talk is that there doesn't seem to be any redundancy. Like when you don't have PRDM9, you aren't positioning the brakes, and that seems really dangerous when you think about the process of meiosis and the fact that you do want to be able to have this process happen and diversity get introduced. Was that surprising to you?
Yeah, well, let me put it slightly differently. There was a big surprise with there is still a big surprise and still an unsolved paradox with this protein.
Okay.
But for a slightly different reason, is that this protein is found in many vertebrates. It's probably, we are not sure exactly the phylogeny, but arose probably with the emergence of vertebrates or maybe earlier, but has been lost independently in many vertebrate lineage. So it has been lost in birds, in some mammals.
That's surprising.
In some fish, and those species without this PRDM9 protein do perfectly well. And recombination takes place at sites which are apparently accessible chromatin, and maybe they have also some histone modification, in particular the one that we work on, the modification of the histone H3.
Okay.
So the pattern of recombination is completely different in mammals, or in some mammals, like in humans, and in another mammal like dog, Canidae, which don't have PRDM9. And so the puzzle is why this, so there are two alternative pathways. That's the way it looks like.
Okay.
And PRDM9 is a very, very specific and unusual pathway, because what I didn't talk about is that its activity destroys its own binding sites across generation. So it's a very puzzling. So why PRDM9 is doing this, whereas other species can do perfectly well without it? Has there been an advantage for this specific function? And what PRDM9 does is that also it makes the position of recombination events dynamic. I mean, dynamic during evolution, meaning that they change over time.
So because it destroys its own site, and then you can't then rebind.
Exactly, and then there are new variants that come in that probably selected and that designed recombination to take place at different sites, whereas species which don't have PRDM9 have fixed and stable sites of recombination. So to answer that, we are, that's why also that's very exciting for me, because we are right at the interface of, to understand this molecular, we need to take into account the long-term evolutionary implication. And I'm now constantly discussing and interacting with evolutionary biologists to try to appreciate this.
Oh, that's
Yeah,
exciting.
that's the challenge.
So on the one hand, you're interacting with evolutionary biologists.
Yeah.
On the other hand, you're trying to understand the molecular interactions that let you bridge from PRDM9 and specific binding sites to SPO11 that's actually
Exactly.
doing the cleavage.
Exactly.
What approaches are you taking to look at that?
For this second part, we need to biochemistry.
Yeah.
I mean, this is, and we are, so that's not easy because the cells that we are working on are mouse oocytes or mouse spermatocytes. But then it's a completely different approaches and different experimental procedure on its biochemistry. So we go now more and more into a biochemical approach.
Mm-hmm.
Yeah.
Yeah, and so, I mean, is it tempting because you are working with mouse spermatocytes or oocytes? It's so hard. Are you bringing in other model organisms as well, or are you sticking with mouse because of the concerns about differences in mechanism?
Well, yes. I mean, that's an important point you raise, is that we are always thinking whether the system we are using is the good one, right, to answer the question. But in this specific context, if we want to understand where PRDM9 and how it works, honestly, I don't have today, I don't know today much which alternative I could take. Okay. There are other organisms where, you know, molecular understanding of recombination is very well developed, like yeast, but yeast doesn't have PRDM9. Mm-hmm. So that's not an option. Mm-hmm. So right now, that's my choice, and we have to continue and use this system if we want to progress. Yeah.
Yeah, and so I'm curious if you think, so those are the questions that you're thinking about now. If you think about what's kind of just beyond reach, like where do you want to be? What questions do you want to be answering in 3 years?
So I think what I've told you really, it's these are events at the DNA level. Yeah. But there is another aspect of meiosis which is fascinating, and that's also part of my interest is I'm interested in the molecular mechanism, but not in all the details. I don't think I will go into in the deepest details of a particular molecular reaction. That's not my interest.
Okay.
I'm rather interested to integrate with how these events at the DNA level take place in the context of the whole of the chromosome of the nucleus. And meiosis, this particular stage, which is meiotic prophase, is really fantastic, and it's plenty of completely unsolved questions, because at the same time these breaks are formed, or before or after, but somehow when the breaks are formed, they have to be repaired and interact with homologous sequences which are present on the homologous chromosomes. And there is, I think, relatively black box about how a broken DNA is going to find the homologous sequence.
I think in textbooks it's always just drawn as, and somehow it's there and it's invading.
So this is, nobody really knows how it works. Chromosomes have to find each other, these DNA sequences have to find each other, the chromosomes have to move, so there is already some data about movement and that. So to answer your question, yes, the idea would be to link this molecular event with chromosome movement and dynamics, and ideally to follow the movement live in the nucleus.
And so to get to the point where you can answer those questions, what are you gearing up to do in the lab?
So for that we need new tools. So I think these questions with the tools that we have now, we cannot really answer. So we have to develop new technologies, or to use technologies that have maybe used for the different purposes, and but to use them for and to design them in this particular system, which is the mouse meiosis. Yeah.
And so if there was one technology that you could just invent, like have at your disposal tomorrow, what would it be?
Well,
or what question would you ask first if you could pluck a new technology out of the air?
Well, I want to follow a specific DNA sequence as the meiosis is progressing, and how the two sequences from two homologous chromosomes, and to follow them in the nucleus during the process of meiotic prophase. And that's not there to do. So that would be a challenge.
Yeah. So I want to take a bit of a step back and ask you. So you've had this fascination with meiosis and both at the molecular level and also thinking about evolution. It's tough to imagine you turning away to work on a different problem, but if you couldn't work on meiosis anymore, what would you work on? If somebody handed you a pile of money and said, go do something new, what would you do?
I think I will, maybe I will still get back to some evolutionary aspects. So then about that I've been thinking about two things. First is to reconstitute an ancestral, the molecular, the proteins from the ancestor from the one that we are studying now.
Yeah,
so
people do this kind of archaeology. It's fun.
Yes, yes. So that's one aspect.
Yeah.
And the other one is similar but in an opposite direction. I would like to take cells, and to have them going for 50,000 generations, and to have a system to. And some people do that, I know, in bacteria.
Yeah.
Because with a generation time of 20 minutes,
you can do those.
You can do that. But I would like to have an evolution to reach the future, right? That would be the experiment.
That would be exciting. Thank you very much for talking with me.
Thank you.
It's been
Thank
fun.
you. Thank you. Have a good day.
You too.
Good.